In this chapter, we'll be taking a look at the senses. I think we can all have an appreciation for our senses, yet their importance extends far beyond deriving pleasure from our environments. In the 1950s, behavioral scientists at Princeton University studied the methods by which the Soviet communists extracted confessions from political prisoners, which included solitary confinement and sensory deprivation. And student volunteers, this would be an interesting research project to be in. Student volunteers were immobilized in dark soundproof rooms or suspended in dark chambers of water. And in a short time, they experienced visual, auditory, and tactile hallucinations, incoherent thought patterns, deterioration of intellectual performance, and sometimes morbid fear and panic. Wow. Well, sensory input is vital to the integrity of our personality and intellectual function. And moreover, some of the information communicated by sense organs never even comes to our conscious attention, such as blood pressure and body tension, or body temperature and muscle tension. These sense organs initiate somatic and visceral reflexes that are indispensable to homeostasis and to our own survival. In this first section, we'll be looking at the properties and types of sensory receptors. And our learning outcomes or objectives are to be able to define the word receptor and sense organ and list the 4 kinds of information that are obtained from sensory receptors and outline a few ways of classifying receptors. Many things that we'll do in this chapter are also covered in lab, so we won't cover much of the end parts in lecture, especially on the specifics of each of the different types of senses like hearing, smell, and touch, etc. But we'll look for the general information at that part of the chapter. So a sensory receptor is a structure specialized to detect a stimulus. Sometimes it can just contain just some bare nerve endings. True sense organs actually have nerve tissue surrounded by other tissues that will enhance the response to a certain type of stimulus. Sometimes those are called accessory tissues, and it can be like epithelium, muscle, and connective tissue. But all in all, receptors are very specific in what they respond to. And the things we'll talk about in this chapter are actually like the beginnings of that reflex arc that we talked about in the previous chapter. And going back to what we were talking about here, for some examples, muscle spindles don't have other tissues. Like, for instance, the eye has a lot of other supporting tissues to help it out. Receptors are actually transducers. A transduction is the conversion of one form of energy to another. And the fundamental purpose of any sensory receptor is to convert that stimulus into nerve signals. The job of a receptor is to turn a stimulus into an action potential. But first, it usually starts with a receptor potential, which is a lot like a graded potential. It's a small local electrical change on a receptor cell brought about by a stimulus. If it's large enough, if it gets to threshold, then it can generate an action potential and then send that to the central nervous system to be transferred into a response. The sensation is an awareness of stimuli. And most sensory signals are delivered to our central nervous system and they produce no conscious sensation at all. They're filtered out in the brainstem and then prevent information overload, sometimes also called habituation. Some signals don't even require conscious awareness, like the changes in pH and body temperature. Thank goodness, because we wouldn't want to be having to worry about that all the time. Sensory receptors can transmit 4 different kinds of information, and we'll talk about each 4. The first one is modality. This is the type of stimulus or the sensation that it actually produces. The type of stimuli they're responding to, like for instance, a light receptor responds to blue light. Then an action potential is generated, and the central nervous system interprets that as what it is, blue light. Location. This is encoded by which nerve fibers are firing, and the brain actually identifying the site of the stimulation. For instance, in a skin demonstration here, we have what's called the receptive field. Some areas have small receptive fields, such as our fingertips, to be able to find two-point touch discrimination, and some areas have a longer receptive field. Areas that don't need the receptors so close together, that don't need the fine two-point touch discrimination. Intensity. This is based on the number of action potentials per second. Just remember, all action potentials are the same size. They're not bigger or smaller, but it can be the number that are occurring per unit of time. And having more per unit of time would make for a more intense response. So for intensity, the brain can distinguish stimulus intensity by which fibers are actually sending the signals, how many fibers are doing so, and then how fast the fibers are firing, aka the number of action potentials per unit time. And then duration, how long the stimulus lasts, so the time that the action potential begins to the time that it ends. All receptors show sensory adaptation, so that if a stimulus is prolonged, the firing of the neuron can get slower over time, and it's classified by how quickly they adapt. For instance, phasic receptors adapt rapidly and generate a burst of action potentials when first stimulated, but then quickly reduce or stop signaling, even though that stimulus still continues. For instance, smell. You probably have noticed when you walk into a new house of a friend or an office somewhere, there's always a new smell that you can associate with that place. But after you've been there for a while, you don't notice it anymore. For instance, you put your t-shirt on this morning, and do you still feel what that feels like? And the answer should be no, because you don't want to have to worry about that. You want to worry about new information coming in, changes, things that could affect your survival. You don't want to bog down the brain with all this constant useless information. Tonic receptors adapt slowly and generate nerve signals more steadily throughout the presence of the stimulus. Proprioceptors are among the slowest because our brain must always be aware of what they control, like body position, muscle tension, and joint motion. So in summary, we're dealing with the receptors. There's first the stimulation of the receptor. Then there's the transduction. That's when it's turned into an action potential. Then there's the conduction. That's when it's propagated to the central nervous system. And then the translation. This is where the action potential is turned into information such as a sound or an image or a taste. Remember that what we see, what we hear, what we feel is actually a bunch of action potentials. Here's how we can classify receptors by modality, and there's some different groups based on modality or based on what they respond to. So for instance, thermoreceptors, they respond to temperature changes. Photoreceptors, like the receptors in your eye, respond to light. Nociceptors respond to pain. Those are pain receptors. There's chemoreceptors. That's basically a protein molecule that binds to a chemical, and then that action opens up a channel, and then that's all it takes to be that kind of receptor. Other chemicals could be glucose, could be oxygen, could be ammonia in the air that receptors in your nose pick up. I would like to mention that nociceptors are actually a type of chemical receptor. They could actually be thought of as that, because what pain really is like the response to when cells are damaged and they break open, spill their contents, which are chemicals, and those receptors pick up those chemicals, generate their action potential, and send it to the central nervous system, which then interprets that as pain information. So that all, though we call it pain, it started with chemicals. Last listed there is the mechanoreceptors. Those respond to mechanical changes or pressure. So for instance, auditory info from your ear is responding to pressure waves, pressure changes in the air, which then is transferred to sound. They can also be classified by the origin of their stimuli. So for instance, exteroreceptors detect external stimuli. Those can include receptors for vision, hearing, taste, smell, touch, heat, cold, and pain. Interoreceptors detect stimuli in the internal organs, so internal stimuli. These are places like in the stomach and in the intestine. And then proprioreceptors sense body position and movements. These occur in muscles and tendons and joint capsules. Then they can also be classified by distribution. The distribution of the receptors in the body, and there's two broad classes of these senses. One is general, also called somatosensory or somaesthetic senses. These employ widely distributed set receptors in the skin, the muscles, the tendons, joints, and the viscera. They include touch, pressure, stretch, heat, cold, pain, as well as many stimuli that we don't actually perceive consciously, like blood pressure and composition, like pH of blood as well. Those receptors are pretty simple, sometimes no more than just a bare dendrite. Then there's the special senses that are limited to the head, and then they're innervated by the cranial nerves, and they employ relatively complex sense organs. These are like vision, hearing, equilibrium, taste, and smell. In this section, you should be able to, when we're done, list several types of somatosensory receptors. Those are from the general senses, also called somaesthetic. be able to describe the projection pathways for general senses, and explain the mechanisms of pain and our ability to block that pain by spinal blocking and pain signals. Receptors for the general senses are pretty simple, usually consisting of some sensory nerve fibers and some connective tissue. We have a range of different receptors to have a range of different responses. We have unencapsulated nerve endings. These lack connective tissue or wrappings. We can usually find these on the surface of the skin or near a hair follicle. They can be just free nerve endings or the tactile discs or hair receptors. Usually they're very sensitive and again near the surface of the skin. Then there's the encapsulated ones. These are encapsulated or wrapped by glial cells or other connective tissues. This wrapping enhances sensitivity or selectivity of response. It might even make them less sensitive, or they may need more pressure to get the same response. Here's some of the different types of encapsulated nerve endings, like the tactile, or if you remember talking about skin, the Meissner corpuscles, the Krauss end bulbs, the Pacinian corpuscles or lamellar corpuscles, and then the bulbous Ruffini corpuscles. And I'll bring us back to this picture here, back to the skin, which you did last semester. Up here, here's some of those free nerve endings that are near the surface and are very sensitive. They don't have any wrappings around them. The Meissner's corpuscles there. Here's a Pacinian corpuscle there and there. They may take a little bit more pressure to get them to respond. They may just respond when the pressure gets more intense, when you want to know that something's going on. The somatosensory projection pathway means the pathway of response from receptor all the way to central nervous system to the correct interpretation of that action potential. So from receptor to final destination in the brain, there's usually 3 neurons involved, called a first order neuron, a second order neuron, and then a third order neuron. So with the first order neuron, from the body it enters the posterior horn of the spinal cord via the spinal nerves. From the head would enter the pons or the medulla via the cranial nerve. And these include touch, pressure, proprioception fibers that are large and fast with myelinated axons, and also heat and cold fibers that are small and unmyelinated. In the brainstem, the first order fibers of these neurons synapse with the second order neurons that decussate and lead to the thalamus. The third order neurons then complete the route to the cerebrum. Proprioceptive signals from the head are the exception to this rule, and as the second order fibers carry these signals straight to the cerebellum. Pain. Pain is any discomfort caused by tissue injury or noxious stimulation and typically leads to evasive action. It's important because it helps protect us to know when damage has occurred, so we take care of it, have the appropriate behaviors. Sometimes nerves or receptors are lost in the disease diabetes mellitus because of diabetic neuropathy. So for nociceptors, there's 2 types, providing different pain sensations. There's fast pain and slow pain. For fast pain, these travel on the myelinated fibers, which are faster. That gives us that short, sharp, localized stabbing pain perceived with an injury, like the pricking of a finger. That's a sharp, localized stabbing pain. The slow pain travels more slowly because they're on unmyelinated fibers, and it's longer lasting and dull with that diffuse feeling. It could be like that headache that won't go away. Both of these can tell the body that there's some damage or some problem someplace. Somatic pain is from the skin, the muscles, and the joints, whereas the visceral pain is from the visceral. Now remember, injured tissues or cells release chemicals that stimulate pain fibers. That's why I had said before that nociceptors are actually a form of chemoreceptors, because they're responding to the chemicals that pour out of these cells that have been damaged. These make us aware of the injury, and then it can activate a cascade of other reactions that would promote healing. And then there's other things that can stimulate nociceptors, like histamine, prostaglandins, and serotonin. There's also another chemical mentioned in your book called bradykinin. It's the most potent pain stimulus known. It hurts intensely when injected under the skin. I guess we want to stay away from that, huh? There's two main pathways to the brain and some multiple subroutes that could be possible. For pain signals from the head, the first order neuron travels to the cranial nerves listed there, and then they end in the medulla. Then the second order neuron picks that up and ascends to the thalamus, and then travels to the third order neurons to the postcentral gyrus of the cerebrum, where then it's interpreted as pain. Pain signals from the neck down travel by way of three ascending tracks. Number one is the spinothalamic tract. It's the most significant pain pathway and carries most of the somatic pain signals. Remember, somatic pain is from skin, muscles, and joints. Then there's the spinal-reticular tract that carries pain signals to the reticular formation we've talked about in past chapters. That activates the visceral, emotional, and behavioral reactions to pain. And then the brachial fasciculus carries signals to the thalamus for visceral pain. Remember, visceral pain is from the viscera, from the guts. And this diagram shows those three different pathways from the previous slides. Either way you look at it, the receptor here picks up the pain information, sends it to the central nervous system by one of the pathways, and then it's perceived as pain. Remember when we talked about the brain, we talked about this area here as our sensory cortex. Well, look how these pathways end here. Sometimes it's really hard to tell, based on where something's hurting, what's wrong. That's because pain in the viscera often mistakenly are thought to come from skin or other superficial sites. This results from the convergence of neural pathways in the central nervous system. For instance, your brain assumes visceral pain is coming from the skin. It can't distinguish the source. For example, when you're having heart pain or a heart attack, where do people say it hurts? Well, they may say their left arm or their shoulder. If we look at this diagram here, we can see why. That heart pain can be felt anywhere in this red area. If, for instance, someone's having kidney pain, look at the yellow area, all the different places that it could hurt if something's wrong with the kidneys. The kidneys are up here, but yet it could hurt all the way down there. Again, that's because of the convergence of the different pathways. person's physical and mental state can greatly affect his or her perception of pain. For instance, many mortally wounded soldiers, for example, report little or no pain. The central nervous system has analgesic or pain relieving mechanisms that are just beginning to be understood. The discovery is tied to the long known analgesic effects of opium and morphine and heroin. Neurophysiologists discovered receptor sites for these in the brain and wondered why we have these receptors since we don't actually have those chemicals, morphine, opium, and heroin in our blood. And so what they discovered is that the central nervous system internally produces opium-like substances. They're called endogenous opioids, and they're divided into a few different groups. Enkephalins are two analgesic oligopeptides with 200 times the potency of morphine. Later, they discovered some other ones that they call endorphins and dynorphins. They're secreted by the central nervous system, the pituitary gland, even the digestive tract, and other organs in times of stress and in exercise, and can act as neuromodulators that block pain and then give pleasure. This could be one of the uh, explanations for what runners call their runner's high, or what they get when they get their second wind. For pain to be perceived, signals from the nociceptors must get beyond the posterior horn of the spinal cord and then travel to the brain. Through mechanisms called spinal gating, pain signals can be stopped at the posterior horn. In the normal pain pathway, we have the nociceptor that is stimulated and then stimulates a second order nerve fiber with substance P neurotransmitter, and then the second order fiber sends a signal up the spinal thalamic tract to the thalamus, and then the thalamus relays the signal to the cerebral cortex where awareness of pain occurs. That's the normal pain pathway. But in pain blocking or in pain modulation, in the posterior horn, descending analgesic fibers synapse on short spinal interneurons, and these interneurons secrete enkephalins, and then inhibit those second order neurons. And some of the fibers from the medulla also exert presynaptic inhibition by synapsing with axons on nociceptors and blocking the release of substance P itself. And this diagram, steps 1 through 8, goes through that pathway in a little bit more visual form. Another way of spinal gating is rubbing or massaging the injury. When you rub or massage an injury, input from mechanoreceptors in the skin and the deeper tissues stimulate the pain inhibition in that posterior horn of the central nervous system. It stimulates those spinal interneurons to secrete the enkephalins that inhibit second order pain neurons. So, see, rubbing and massaging an injury does help. This is the part where I will be shortening it a lot because you will cover a lot of these other special senses during lab. So I'm only going to briefly go over just a little bit about each of our special senses. Taste is also called gustation, and it begins with the action of chemical stimulants on the taste buds. Those are called tastants. There's about 4,000 found mainly on the tongue, and especially in children, they have a few more inside on the cheeks, the soft palate, the pharynx, and even the epiglottis. There's 5 primary sensations that we can experience. They're salty, which is produced by the metal ions sodium and potassium. Sweet, associated with carbohydrates. Sour, like the acids in citrus fruit. Bitter, like spoiled foods and other alkaloids like nicotine, caffeine, quinine, and morphine. And then umami, which is actually that meaty taste of all the amino acids in chicken or beef broth. Remember, for any of these senses, it's the receptors picking up that information and creating action potentials sent to the central nervous system and then perceived as the right information. The next one, smell, also called olfaction. It's the response to chemicals in the air or odorants. Our olfactory mucosa can contain up to 10 to 20 million olfactory cells or neurons. And our mucosa of the superior concha, nasal septum, and roof of the nasal cavity cover about 5 centimeters squared. On average, we have about 2,000 to 4,000 odors that we can distinguish. There's people out there in the world that are called super smellers that can actually smell more. All in all, though, when compared to other organisms, other mammals, humans are said to have a poorer sense of smell than most other mammals. But still, it's even more sensitive than our sense of taste. And women tend to be better at smelling odors than men, especially at certain times when they're ovulating. Humans have only about 350 types of olfactory receptors. Mammals that have better sense of smell will have more. For human pheromones, we've often wondered how much do we actually respond to the smells that come off of our body. Well, it's been shown that human body odors can affect sexual behavior. A person's sweat and vaginal secretions can affect other people's sexual physiology, and they talk about the dormitory effect when women live together, that all of their menstrual cycles can actually sync up with each other because of the smells that are released. And the presence of men seems to influence female ovulation. Ovulating women's vaginal secretions contain pheromones called copulines that have been shown to raise men's testosterone level. Remember, in detecting different smells, an action potential has to be generated. Receptors in your nose monitor the air, and the receptors respond to the different chemicals. Those chemicals bind to the receptor and then generate the action potential, which is sent to the central nervous system and then perceived as an odor. For hearing, it's a response to vibrating air molecules, and equilibrium goes hand in hand with hearing. It senses the motion of the body, the orientation, and the balance. And both of these senses reside in our inner ear, which is a maze of fluid-filled passages and sensory cells. The fluid in your inner ear is set into motion, and the sensory cells will convert this motion into an informative pattern of action potentials. A sound is any audible vibration of molecules. Those air molecules will hit the eardrum and cause it to vibrate. The ear has 3 sections, the outer, middle, and the inner. The first 2 are the ones concerned only with the transmission of sound to the inner ear. In the inner ear, that's where the vibrations are actually converted to nerve signals. I'll get back to that in a second. First of all, though, pitch is actually our sense of whether something is high or low, which is determined by the wave's frequency measured in hertz, so the number of cycles per second. So, for instance, if you wave your hand back and forth, you could call that cycles per second, but it's going so slow that you cannot hear that. But if you could get it up to 20 per second, perhaps you'd be able to hear it. You can hear up to 20,000 cycles per second, or 20,000 hertz. Infrasonic is below 20, ultrasonic above 20,000. Some organisms can hear ultrasonic and infrasonic, meaning above and below what humans can hear. Speech is about 1,500 to 5,000 hertz, which is our best areas of hearing where we're most sensitive to that. And most hearing loss with age is a range of 250 to 2050 hertz. Loudness is the perception of sound energy, or the intensity or the amplitude of the vibration. We express it in decibels, and always want to remind everybody that anything above 90 decibels can cause permanent damage. So, the vibration of ossicles, that's the inner ear bones, causes the vibration of the basilar membrane under the hair cells, as often as 20,000 times per second. And then those hair cells will move with that bottom membrane. And as they move, in this picture, these are depicted as the cilia, or the hair-like structures. As they move or as they tip, this opens up channels and potassium floods in, and the action potential is generated. And then we hear sound, or it's interpreted by the central nervous system. Deafness is hearing loss. Conductive deafness are conditions that interfere with the transmission of vibrations to the inner ear. And nerve deafness, or sensorineural, is death of hair cells or any other nervous system element concerned with hearing. For instance, musicians that have been exposed to loud music over time, or factory and construction workers who are in the presence of loud sounds and don't use hearing protection, can fall ill to nerve deafness. The last one we'll talk about is vision. And I might go a little bit more in depth with this one than I have with the other ones. But lack of time, I'm not going to go too much into everything with all of these senses. But I do always encourage you to definitely read your chapter, and there's a whole wealth of different information, somewhat even to the neurosurgeon level, that we won't get to here. But we'll do some of the general stuff. So vision or sight is the perception of objects in the environment by the means of the light that they emit or reflect. So light obviously comes from the sun, what we call the electromagnetic radiation, and there's different wavelengths of radiation coming down and hitting us. And in the wavelength of light from 400 to 700 nanometers, that's what we say is the range of human vision, also called the range of white light. Below 400 nanometers is the ultraviolet radiation. This has too much energy and destroys macromolecules, as seen in tanning beds, when exposure to this can destroy DNA, cause cancer. Infrared radiation is said to be above 700 nanometers. Here there's too little energy to cause a photochemical reaction in the receptors in our eyes, but we do know it's there. We can feel it warm our tissues. And I've included this diagram of the anatomy of the eye. There's three principal components of the eyeball. The optical components emit and focus light, and the neural components are the retina and the optic nerve, and the tunics form the wall of the eyeball, which consists of 3 layers. Here's a neat picture of someone using an ophthalmoscope, listed right here, to see the retina of the eye. The macula lutea is the patch of retina on the visual axis of the eye. The fovea centralis is the center of the macula. It's finely detailed images because there's a lot of receptor cells there. So they're talking about this area right here on the retina, the fovea centralis and the macula lutea. Over here is the optic disc. This is where your optic nerve actually leaves the eye. So you don't have receptors right here. So that's actually referred to as your blind spot. You can do a neat little trick. If you close your right eye and stare at this X and move your head closer to the computer screen or farther away, you'll see a point off in your peripheral vision where this red dot disappears. And that's called finding your blind spot. So that red dot is in the field of vision of where your optic disc is at, where there's no receptors. You might also notice the visual filling that your brain actually fills in the green line for you. Even though it's not seeing anything there, it will assume and then fill in the rest of the green bar. Some problems with eyes are cataracts and glaucoma. A cataract is the clouding of a lens, sometimes with age, sometimes with lifestyle choices like smoking and tanning, being in the presence of ultraviolet radiation, and so on and so forth, or other disorders. You can treat it by replacing your natural one with a plastic one. There's glaucoma, which is elevated pressure in the eye due to an obstruction of the scleral venous sinus that would drain the aqueous humor in the eye. That would be the fluid. It can cause death of your retinal cells due to compression of those blood vessels and then deprivation of oxygen. Some symptoms are some illusionary flashes of light as an early symptom. Colored halos around the lights are late symptoms. And once that vision is lost, when you've made that damage on that retina, it cannot be restored. Pressure is usually measured at the eye doctor's office with a tonometer. In the formation of an image, light must pass through the lens to form a tiny inverted image on the retina, and the retina must convert that light energy into action potentials for your central nervous system to perceive as light or an image. To help that out, here's the structure of the retina on this slide. And we'll look at a picture of this when we're done. At the very back, the most posterior part is the pigment epithelium, with pigment back there to absorb stray light so that visual images are not degraded. Then there's different neural components. From the rear forward, there's photoreceptor cells, which is kind of interesting to have the actual cells that receive the light way in the back, and then those are the ones that generate a chemical or electrical signal or an action potential. These consist of a few different types called rods and cones usually. Then there's a little bit further yet forward or more anterior are the bipolar cells. These are first order neurons. They have dendrites that synapse with rods and cones and axons that synapse with ganglion cells. Then come further forward, we have the ganglion cells, which are the second order neurons, and they're the largest in the retina and are the ones that are going to send the information to the optic nerve. So you send the light to the back of the eye, and then that message actually transforms further forward into the eye and then back again through the optic nerve. A little bit more on the rods and cone cells here. Those are the light absorbing cells shown in the pictures here. They have some similarities and differences. They derive from the same stem cells as ependymal cells of the brain. Rod cells are sometimes called for scotopic vision or night vision, and or also monochromatic vision. The outer segment has modified cilium to absorb light, and you can stack 1,000 membranous discs studded with globular protein. The visual pigment is called rhodopsin, sometimes nicknamed visual purple. The inner segment contains organelles sitting atop the cell body with the nucleus. The cone cell, called the photopic cell or color cell or for day vision, is similar to the rod cell except that the outer segment tapers to a point up here. And the disc with pigment are plasma membrane infoldings. They're not detached. There's that picture. You have about 130 million rods and 6.5 million cones in the retina. Only 1.2 million nerve fibers in the optic nerve, though. But with neuronal convergence and information processing in the retina before the signal reaches the brain, multiple rod and cone cells can synapse on one bipolar cell, and multiple bipolar cells can synapse on one ganglion cell. So way back here, here's that pigment epithelium that I talked about in a couple slides ago. Then the rod and cone cells, this is where the light is actually detected. And then that message is sent forward in the eye on these bipolar cells here to the ganglion cells here, which then send the action potential to the optic nerve. There's some other horizontal cells here that help tie it all together. Rhodopsin or visual purple is one of the visual pigments. There's two major parts of the molecule, the opsin, which is the protein portion embedded in the disc in the rod's outer segment, and then the retinal is how it's said, or retinine, which is a vitamin A derivative, which is why it's important to have your vegetables that have vitamin A in it. It has an absorption peak at about 500 nanometers. Rods, though, cannot distinguish one color from another. Cones have a pigment called photopsin or idopsin. Retinal moiety is the same as the rods. The moiety is like a mixture. The opsin moiety or mixture contains different amino acid sequences and then therefore would respond to different wavelengths of light. Then the three kinds of cones are identical in appearance, but they absorb the different wavelengths of light to produce our color vision. There's some pictures of the pigments. When light hits it, it actually changes form, and your body has to change it back. That's called bleaching. So it takes time to do that. That's why it takes time to adjust when you turn on the light in the morning or to adjust to night vision at night. So to generate the optic nerve signal, the bipolar cells will detect fluctuations in light intensity and will stimulate the ganglion cells directly or indirectly. The ganglion cells are the only retinal cells that produce action potentials, and they respond to the bipolar cells with rising and falling firing sequences or frequencies. Then via the optic nerve, these changes provide the visual signals to the brain. So remember, we went from the rods and the cones to the bipolar cells, then to the ganglion cells, and now to the optic nerve, to the central nervous system, to the brain. So this slide talks a little bit about what I've already mentioned here with the light adaptation and the dark adaptation. How it takes a little bit of time to regenerate your pigments in the rods and the cones to be able to work at their optimum sensitivity. Primates have well-developed color vision, whereas nocturnal vertebrates only have rods so that they can see well in the dark. We have three types of cones, sometimes called short wavelength, or S cones, medium wavelength or M cones, and long wavelength or L cones. But sometimes they're called blue, green, and red cones. Not that they only see those colors, but it basically means where they peak in the color that they see. Here's where the rods peak. This is where the S cones peak. The blue cones and the green cones are M cones, and the L cones are red cones. And so from here to here, the wavelength of the rainbow of light is the visible spectrum for humans, and a mixture of how much each of these cones are stimulated determines the color that we see. For color vision, there's a sex-linked recessive disorder called red-green color blindness, which is more common in men, where they lack um, one of the different pigments or the functioning of their cone cells. So if they were to look at this picture here, they would not see that number in there. If you're looking and you have good color vision, you should be seeing the number 74 in that circle. So these people might lack an L or an M cone, because those proteins are not made with that recessive disorder. It's a DNA gene mutation. With stereoscopic vision, that's another thing that we mammals can enjoy, is being able to see things in stereo. Because of having two eyes, we have depth perception. We can judge the distances to objects by looking at an object with our two eyes. Because those eyes are at different angles, our brain can judge distance to something. So you can be standing and somebody standing a little further from you, and you can fixate on them and throw a ball to them and judge that distance just by looking at them. The visual projection pathway of sight. So our rods and our cones are the receptors. The bipolar cells of the retina are the first order neurons. The retinal ganglia cells are those second order neurons, whose axons then go on to form the optic nerve. The two optic nerves combine to form the optic chiasm. Half of the fibers will cross over to the opposite side of the brain. That's called hemidecussation. And the chiasm splits to form the optic tracts. You can see that in this picture right here. See how some of the fibers go all the way to this side and some go to that side? The right cerebral hemisphere then sees objects in the left visual field because their images fall on the right half of each retina, and each side of the brain sees what's on the side where it has the motor control over limbs. So it only makes sense when there's this crossover. The optic tracts pass laterally around the hypothalamus, with most of their axons ending in the lateral geniculate nucleus of the thalamus. Third order neurons arise here and form the optic radiation of fibers in the white matter of the cerebrum. They project to the primary visual cortex of the occipital lobe, where the conscious visual sensation occurs. And I've included this diagram to show where those areas are, all the way to the visual cortex of the occipital lobe back here.